happening now on The Rundown.
This is one of the most infectious agents we've ever encountered as a species, and I think the implications of that are still sinking in.
COVID-19 is tearing apart the world as we know it, and to contain and control the deadly virus, Montana scientists are in a race against time.
It's a race against time, but it's also a race to make sure we develop something that's safe and effective.
I'm Jackie Coffin, and I'll take you to the front lines of Montana's COVID research.
People, I think, don't realize how cutting-edge research in Montana is.
I think it's very gratifying that a little lab in a little corner of Montana is playing such a major role.
And give you a look at my own experience with the search for the cure.
Vaccines are really the most effective medical measure we have, like, period.
The Rundown starts now.
A dangerous virus is spreading rapidly in China, and U.S. officials are very worried that it could come here.
As COVID-19 grew globally, spilling into every country and moving towards Montana, scientists in every corner of the state readjusted their sights on the new pandemic, trying to figure out what it was and how to stop it.
The people that say no one saw this coming, that's nonsense, because a lot of people saw this coming.
They're waving the red flag, but nobody's listening.
As an expert, we know it would be coming, but you're still, obviously, still surprised when it happens.
As a person and a journalist, I love and am fascinated by science. I grew up in Montana's scientific community. My dad is a professor of molecular genetics, and my stepmom is a professor of neuropharmacology, both at the University of Montana. And as COVID-19 hit, spread gradually, and then like wildfire, it felt natural for me to look to Montana's scientific community for answers.
There's no question Montana scientists are at the cutting edge of this.
And this took me down a thrilling path on the search for the cure.
To whip this pandemic, we need three things. We need antivirals, so drugs that will eliminate the virus. We need vaccines, and we need better diagnostics.
Detection, vaccines, treatment. Detection, vaccines, treatment. These are three of the main boxes Montana scientists are trying to check with hundreds of hours in the lab, because the three together lead to the ultimate goal: prevention.
Everyone has a sense of urgency that we need to get this virus under control.
It's a race against time, but it's also a race to make sure we develop something that's safe and effective.
Before we get too deep into any of the work being done, let's assess what we're dealing with. First, what is a virus? A virus is a microscopic pathogen that is constantly searching for a host to replicate in and take over. Viruses cannot grow without a host because they're not even a complete cell. They're just a tiny little package of nucleic acid in either DNA or RNA. Every living thing in the world has DNA and RNA. DNA writes our genetic code, and RNA translates that code into action. Whether viruses are alive is kind of a gray area. While they contain some of the elements that constitute life, like DNA or RNA, they can't do anything with it unless they find a host organism, a plant, an animal, a person, and use our cells to replicate itself, which ultimately destroys the cells. So instead of thinking of viruses as little creatures, it's more like a microscopic android trying to take over our cells. One host is not enough for a virus, and it will spread to host after host after host after host, maybe through a sneeze, a cough, a simple touch, or even inhaling and exhaling. Using saliva and water droplets as its mode of transportation, and the mouth, eyes, nose, and respiratory system as its way into a new body. When a virus travels this way, it's aerosolized or airborne. And the way to block it from spreading host to host is covering coughs and sneezes, washing and sanitizing hands and shared surfaces, and blocking its entrance into the new host's respiratory system using masks or face shields. Not every virus can travel this way. Ebola and HIV travel through direct contact with infected blood and bodily fluid. As we all know, SARS-CoV-2, a coronavirus, can, and that's what we're dealing with now. To touch on taxonomy, SARS-CoV-2 is the name of the virus that causes the disease, COVID-19. But where did it come from? How does a virus manage to infect 72 million hosts in every country and territory in the world? We can find the answers in Bozeman.
Viruses exist everywhere, just like bacteria. They're in the air, on surfaces, in water, everywhere.
A virus can't run, it can't walk, it can't fly, it can't crawl. They don't come after us, they fall into us. And they generally fall into us because of things that we do more than things that they do.
Digging deep down to the roots of global pandemics and infectious disease are David Quammen and Devin Jones. Jones is a PhD candidate at the Bozeman Disease Ecology Lab run by Dr. Raina Plowright on the MSU campus.
I ended up here because I was really interested in bat health. I studied the gut microbiome of flying foxes in Australia.
David Quammen is a renowned science journalist and best-selling author who has tracked infectious disease for more than 20 years.
I have lived in Montana since 1973. I travel a lot, usually, to do research for my books and articles. I worked a lot for National Geographic magazine for 20 years. I've worked for other magazines, the New Yorker magazine, Harper's Magazine, Outside Magazine. Again, mostly but not entirely on scientific subjects. I, for many years, covered ecology and evolutionary biology. And then about 20 years ago, I got interested in emerging viruses, scary new viruses that come out of wild animals. We call those zoonotic viruses. I got interested in them because I was walking across the Congo on an assignment for National Geographic with an American explorer conservationist. And we walked through Ebola territory for a stretch of 10 days. And so I got interested in the mystery of the reservoir host of Ebola. I realized quickly that this is a matter of ecology and evolutionary biology. I thought, oh, okay, I get that. I'm going to write a book about the ecology and evolutionary biology of scary viruses, including Ebola. So I did that. It was titled Spillover, published in 2012.
To source the origins of a global pandemic, Jones and Quammen say you need to understand a process called spillover.
Spillover is the movement of a pathogen from one animal into another. So that can be an intermediate host. So like with SARS-1 in China, we see that that coronavirus moved from bats into civets and then from civets into humans. So those were multiple spillover events. So it could spill over into civets and then spill over into humans. So our lab studies the process of spillover. So what are those drivers that make spillover occur in the first place? And there are a lot of barriers that a pathogen has to go through in order to spill over into a new host. And so we study those different layers and how those layers either align or don't in order for that pathogen to spill over.
SARS-CoV-2 spilled over from bats, a frequent mammalian host to viruses.
Bats seem to carry more than their share of viruses that can get into humans and cause trouble. There's a whole sequence of dangerous viruses that have come from bats as the reservoir host, the species in which they live when they're not infecting humans. And this virus is only the latest of those. Probably Ebola, we don't know that for sure. Marburg virus, Nipah virus in Malaysia and Bangladesh, Hendra virus in Australia, the original SARS virus coming out of China in 2003, others as well. What is it about bats? Well, first of all, bats are a very, very diverse order of mammals. 1 in every 5 species of mammal on Earth is a species of bat.
But bats should not be blamed, say Quammen and Jones. They're losing habitat to development and deforestation, and we, humans, are putting ourselves right in the path of wild animals and new viruses.
Emerging infectious diseases are, at this point, mostly zoonotic, and they're spilling over more frequently over time. And so we think that most of that is due to land use change. But yeah, so there are teams of scientists like in PREDICT in California and EcoHealth Alliance researchers, for example, have been studying coronaviruses in bats for a long time, like decade plus. And they have previously identified coronaviruses as a high-risk virus with pandemic potential. And so there's kind of they're waving the red flag, but nobody's listening. And so then when it happens, people are like, oh, suddenly it's really important to study these bats and their ecology and things like that. And at the end of the day, I think too, it's really important to note that bats aren't the problem, but people have sort of invaded their space and taken away their resources to where they have nowhere else to go and they have nothing left to eat. And so that causes like nutritional and extra reproductive stress that might increase viral shedding, pathogen shedding, and increase the likelihood of these sort of zoonotic transmission events.
Bats carry viruses. How do those viruses get into humans? It's not that because bats are looking for us. It's not because those viruses are looking for us. Neither. It's because we go looking for them. We invade bat habitat. We disturb bats in various different ways, cutting down forests, harvesting guano from caves. If we leave those trees alone, if we leave those forests alone, if we stay away from bats, then bats will be better off and we will be better off too.
As for Quammen's 2012 book Spillover, it is eerily prescient 8 years later.
And at the end of that book, because I was listening to some very smart disease scientists, I put together what they were saying in answer to my questions and essentially predicted COVID-19. They predicted COVID-19. I asked them, is there a next big pandemic coming? They said, yes. What does it look like? Well, it'll be caused by a virus, new to humans, coming out of a wild animal. What kind of a virus? Well, one that evolves relatively quickly, such as an influenza or a coronavirus. What kind of a wild animal? Well, one that's known to carry viruses that can infect humans, very possibly a bat. Where might this spillover happen? Where might this pandemic begin? Well, possibly in or near a wet market or someplace where wild animals are exploited for food. For instance, in China. So that was in the book in 2012. And as a result of that, I've been getting a lot of calls this year.
Now we know what a virus is, how it infects a host, and how it turns from something carried by a bat into a widespread human pandemic. So how do we stop or at least control it?
We need three things.
Detection, vaccines, treatment.
Diagnostics that aren't, multiple day turnaround time. We need diagnostics that are cheap, accessible, point of care, easy to use. All of those things in one convenient package. And we're trying to deliver on that.
On the MSU campus in Bozeman, we enter the labs of Dr. Blake Wiedenheft and Dr. Seth Walk. Both Dr. Wiedenheft and Dr. Walk are finding ways to trace COVID-19 in individuals and in the community through the gut.
When the COVID pandemic started, we got involved in a couple of different ways. The first thing that we started doing was testing wastewater for the prevalence of SARS coronavirus. And the reason that we got interested in that is that there was an early report that not only is SARS coronavirus transmitted through aerosols, and thus the importance for face coverings, but it's also transmitted in feces. Wastewater provides a health record of the community in real time. So with a single test, we can get a sense for what the prevalence of the virus is in the community at any time, and we can monitor that over time. Is it going up or is it going down? And the results from those wastewater tests mirror what's happening in the community, but they do so in a way that provides us that information before we can get it any other way.
We began developing some protocols early on in the pandemic in the US here around May. There was a push to look for SARS-CoV-2, the virus that causes COVID-19, in wastewater. And we collaborated with Blake to get those protocols up and running at Big Sky, in West Yellowstone, and then later on at Three Forks. And we did some research in the lab to try to make the protocols better and more sensitive. We developed some techniques there. And so we had tested throughout the summer, doing weekly testing at those sites. and reporting back what the levels were or whether the virus was even there or not. And that kind of helped public health and local county health to understand how the disease was getting around and being transmitted and where the hotspots were in the county.
The protocols for testing samples established in Dr. Walk's lab came in another use when the state reached out for help processing the immense amount of swab samples starting to roll in mid-summer.
The National Guardsmen and women, they courier samples to MSU every day from Tuesday to Saturday, and we run the tests and report the results back to the state. And over the time, we were able to ramp up our capacity to the point now where we're testing over 2,000 samples a day. It's something about a third of the samples in Montana get tested right here at MSU. That's pretty cool.
Access to widespread testing was an early failure of the U.S. COVID-19 response. Now, drive-through testing is being done at hospitals, fairgrounds, college campuses, including MSU and UM. But better diagnostics are still in demand.
Vaccine's coming, it's getting rolled out, but we still need to be aware that if folks are symptomatic or they're asymptomatic but test positive, they need to isolate, they need to quarantine to really stop that transmission chain between individuals. So detection is at the heart of that. It's the first thing that we can do, but it's really that, what result or what that result triggers, what activities and actions. And that's really what stops the disease and lets us bend the curve or flatten the curve, as they say.
The research institutes we are visiting in this program are not limited to any specific aspect of COVID-19 research between detection, vaccines, and treatment. All of these research institutes feature a wide array of work being done by a lot of scientists, too much and too many to fit into one program. 126 miles north of Bozeman, all three of our silos of prevention are under the microscope at McLaughlin Research Institute in Great Falls.
The McLaughlin Institute is a private, nonprofit institution. It's been in existence since the late 50s. As a relatively small nonprofit research institution, the strengths of the Institute include the fact that we can be very nimble. We were, of course, like scientists all over the world, very interested in helping and trying to do our part in finding, developing technologies and helping the state of Montana develop the infrastructure to meet the requirements for this challenge. Things like increasing our testing capacity, being able to do research in developing vaccines and potential drug therapies for the disease, for COVID-19. We do a lot of genetic work with mice.
An important thing to discuss in the role of scientific research is the use of animal models.
Mice have been, over the past decades, have evolved to be arguably the most important model organism that kind of bridges this gap between basic research at the bench before it can be brought into clinical trials in humans. Mice serve as an excellent model because although obviously they're a very different organism, we share an enormous number of genes in common.
In animal models, scientists are able to replicate the virus and keep up with it in real time. As the virus mutates and new forms of detection, vaccines, and treatment are tested for effectiveness. At McLaughlin, Dr. Kavanaugh took me on an immersive tour to see their work in progress. I went into labs, I went into the main mouse rooms, I met scientists working on editing mice genomes to create a mouse model on the cutting edge of COVID research. New vaccines come with a lot of questions. Will this work? Is this safe? What will it do to my body or to my kid? Who wants to be a guinea pig for some new science experiment? Well, I do.
Comes a little poke.
On September 4th, I got up early and drove from Missoula to Bozeman, jumping at a last-minute chance to participate in the Pfizer vaccine trial. Pfizer, a US-based pharmaceutical Goliath known for creating Viagra, is one of the leaders in developing a COVID-19 vaccine and was in need of 44,000 human participants to sign up to try out the experimental vaccine in Phase 3 human trials. This includes about 100 Montanans in a trial being conducted out of Bozeman Health. To my knowledge, it is the only COVID-19 vaccine study happening in Montana. To qualify for the study, I had to be in relatively good health, not be immunocompromised or pregnant, and not participating in any other types of trials. Check, 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 check. The study is a 2-year commitment that includes 6 visits. 5 of the 6 visits are blood draws, which monitor the level of antibodies the vaccine creates in my blood and how long they last. These are some of the main questions Pfizer is trying to answer in this study. Is this vaccine effective in preventing COVID-19? How long does it take to work? And is it safe once it's actually in humans? My first visit was a blood draw and my first shot. An important note is that Pfizer's study is a 1-to-1 double-blind study, which means that for every 1 vaccine administered, 1 saltwater placebo is administered as well. Double-blind means neither I nor the physician giving it to me knows what I got. Bozeman Health and the vaccine research team gave me permission to shoot this video. I might not look. I do a little bit better with shots if I don't look.
I'm going to ask you not to look because Pfizer's very particular, even though this is totally masked.
Oh, right, of course.
They don't want you to get even the least clue what you've got.
What I have.
So, comes a little poke. Put a Band-Aid on that.
Oh, that was easy. Much easier than my blood draw. Very different outcome.
Well, I will let you know if you do happen to have any sort of symptoms or side effects from your injection. We've seen them with both placebo and active, although on our side, we don't know who has, or we don't know who is experiencing what.
Right.
Give us a call right away.
Okay.
So we want to track that, as your research methods class would dictate, that we want to track that data and that side effect profile for this. But we also want to make sure that we can put you in touch with providers and get you the care you need if there's anything concerning to you. So.
Okay. I kept a video log of my experience as it went on. Hello everyone, I want to do a 2-week update since I got my first dose of the Pfizer COVID-19 trial vaccine. I feel fine. I've been asked that. I've had no reactions except for a little bit of itching on my forearms that happened within a couple of hours after I initially got the shot. And I don't know if that was an allergic reaction to the shot or something in my environment or any matter of things, nothing concerning to me, nothing I was worried about. So I am sitting in my car outside of Bozeman Health Hospital. I just got the 2nd round of my COVID-19 vaccine in this arm, same arm I got the first shot in. It's a booster, 3 weeks to the day after I got my first vaccine. A booster is part of RNA vaccines. It's required if this vaccine goes to market. Other people will have to get boosters of it too. So I'll just tell you a little bit about the process. This appointment I just had was only less than an hour long. Unlike my first appointment, they didn't take all my specs, draw my blood. This was not a blood draw appointment. It was just a booster. The second vaccine, a lot of questions I've been getting about it is, after I got my first vaccine, how do I feel? If anything happened to me, I feel great. My next appointment is in a month and I will be getting a blood draw. And the next 3 appointments after that will be blood draws too. All the rest of the ones will. And all it's looking for is COVID-19 antibodies in my blood to see if these vaccines were effective in building antibodies. One thing about that is, even if I do, is it enough antibodies to prevent me getting sick with COVID-19? That's one of the big questions with these vaccine trials. Still a lot of information we don't know, but we're learning as it goes. So I'm here waiting to get my COVID test, sitting outside in the parking lot in my car. On Friday, today's Sunday, on Friday I started feeling like a little cruddy, sort of along the lines of maybe seasonal changes. It's fall here in Montana, so now it's cold, and I get a cold this time of year pretty frequently. Then yesterday morning, Saturday, I woke up
And I didn't realize it at first, and I was drinking a cup of coffee and I thought, this coffee has absolutely no flavor. And that's kind of when I realized I didn't have any, I had like lost my whole sense of smell and taste. So I went and brushed my teeth and I couldn't taste the toothpaste, and then all day I just couldn't smell or taste anything. So I figured I'd come and get tested because although I'm congested, I can still breathe through my nose, so losing my sense of smell and taste was very strange and concerning to me. I don't know if I've been exposed to anyone with COVID, not to my knowledge, but I travel a lot for my job. You know, I go to grocery stores and my office is on campus, so could be anywhere. It could be an exposure from anything, or it might just be a cold, and hopefully my test results come back negative, because that would be great, but I'll let you know. So I just got off the phone with the clinic and I did test positive for COVID. I don't know when and where I contracted it, and they told me they're going to contact me in a couple of days to begin contact tracing. So an important part of this vaccine study that I've been participating in is recording all of my symptoms or lack of symptoms on a weekly basis and on an app in my phone, as I've shown you guys. And as soon as I reported any COVID symptoms, they immediately called me, and I've been checking in with the research team every single day, and they're asking me how I'm feeling. But one thing they really want me to do, even though I released my positive COVID test results to them, they really want me to self-swab myself with this kit they sent me. So there's a swab right here, and there is this little vial of fluid that will preserve my results, and it has a barcode on it. And then I have my home self-swab requisition card. I have my biohazard specimen bag, and they're gonna come pick this up as quickly as they can, which will in this case be tomorrow morning. This is the self-swab. And I already called them a little earlier. They'll pick it up at 10 a.m. tomorrow.
Fortunately, I had a case that did not turn into a hospitalization or death, and I feel really lucky for that. Within 2 weeks, my symptoms cleared and I was released by the health department. I had a follow-up with my vaccine team right away and had an additional blood draw added to my visits. Getting sick with COVID-19 made me assume that I got the placebo, and that's what I still think. But it's also possible that I did get the vaccine and that I still contracted the virus. Maybe I was exposed too soon after the shot, and maybe it made my case less severe than it would have otherwise been, but that's impossible to know. The first shipment of Pfizer vaccines arrived in Montana on December 14th, and later that afternoon, I witnessed the first doses being administered to 6 healthcare workers at Bozeman Health. And honestly, I felt really emotional.
Receiving the first dose is Dr. Andrew Sullivan, one of our pulmonologists and intensive care physicians.
A year after SARS-CoV-2 spilled over, wreaked havoc, and changed all of our lives, exhausted and relieved healthcare workers are getting their first shots.
My name is Eric Lowe. I'm the medical director for the emergency department at Bozeman Health Deaconess Hospital, and I've also been working on the incident command team here throughout the COVID-19 pandemic. Today was a very exciting day. As a hospital system, we received our first doses of vaccine, the Pfizer-BioNTech COVID-19 vaccine, and we were able to administer our first 6 doses this evening. Emotionally, it felt fantastic to see this finally come to fruition. It's been a long year. I think for everyone everywhere it has, but speaking for the emergency department in particular, it's been a tough year. Dealing with constant change and uncertainty and the unknown and the fears that go along with that are grinding, and to do that day in and day out for month upon month without a clear end has made for a very long year. Well, I think this is something that really should be celebrated. To think about how many people's efforts went into getting to this point is kind of daunting. From the scientists to the airlines that were delivering this, to the delivery people, to It really took all of us to get to this point. It's felt like we're in a long dark tunnel, and now it feels like there's a light at the end of that tunnel.